Hello everyone, my name is Nate. I own a website called MCAT. This is going to be a pretty quick summary of aerobic respiration. So we're not going to talk about literally every single thing that you need to know, just going to be introducing all of the important stuff. You can always go back and watch through my longer videos about each of these processes for more information. I also recommend checking out my website, which is linked in the description. That's where you'll find all of my other free resources. I have all of my videos on there. I have a whole bunch of cheat sheets. I have my own Anki decks that you can use. I have the other really popular Anki decks linked as well. So highly recommend checking that out in case you're looking for some other help for the MCAT. I also offer private tutoring in case you're interested in that as well. The description is where you'll find the link to my website. It's also where you will find the link to the simplified notes that support the info in this video. Since this video is mainly just a summary, the only thing that's really included in today's notes are mostly just the graphics that I use, and you can obviously use those for whatever you want. So to begin giving an overview of aerobic respiration, we have our lovely little stick figure human over here on the right that we're not really going to be doing anything with. I basically just want to start off by asking a few fundamental questions in order to understand why we even do this process in the first place. We're going to start off with whenever we eat a meal. Whenever we eat a meal, we are going to have a ton of glucose in our blood. So what are we going to be doing with it? And the answer to that is we're going to be turning it into ATP. Our cells are always going to need ATP for something, whether it's like fighting an immune reaction, growing, building new proteins, something like that. We always need ATP. So that's what we're going to start off doing. So why do we do that? Specifically with glucose, the answer to that is that glucose contains a lot of potential energy that we can use to actually form ATP. And ATP is going to be the universal molecule of energy that every single organism is going to use. So how do we do that? First, our body is going to rely on aerobic respiration, this process that we're talking about today. This is the way that we're going to convert glucose into ATP. So if we have a whole bunch of glucose in our blood, how do we actually get that glucose into the cells? We have these transmembrane proteins called glucose transporters. They're going to respond to insulin, which is going to ship glucose from the blood into the tissues. So what all is it going to be doing? It is the hormone, it's a peptide hormone, that is going to be removing glucose from the blood, like we just talked about. So it's going to be responding to high blood glucose concentrations. It's going to be telling our cells to either use this glucose to turn it into ATP, or if we have enough ATP, we're going to store it. Basically, insulin is getting glucose out of the blood. So in addition to removing it from the blood, insulin can also directly promote a few different enzymes in aerobic respiration as well. So basically in this fed state where we have a whole bunch of blood glucose, insulin is going to be released just to get it out. So what's going to happen if we don't have oxygen or we need ATP super fast? In this case, we're going to swap to anaerobic respiration. Aerobic respiration is going to be occurring whenever we do have oxygen and we don't necessarily need ATP super fast. That is the main way that we're going to be utilizing glucose to produce ATP. But if we can't do that because we don't have oxygen or our need for ATP is super, super quick, we're going to swap to anaerobic respiration. We'll talk about that a little bit later. So then finally, what happens when we have enough ATP? Then we're going to turn around and instead of utilizing glucose, sending it through aerobic respiration, we're going to store it. This is a process called glycogenesis. There's obviously a separate video dedicated to glycogenesis, but we'll also kind of talk about all of this again in the postprandial / fed state summary as well. So just to give you an overview of what's going on in the cell, specifically eukaryotic cells, we'll talk about how prokaryotes are a little bit different later. So within this eukaryotic cell, I'm going to throw up all of the not really important organelles for the purposes of metabolism. But we're going to start off with talking about the bloodstream over here. So in this fed state, when aerobic respiration is going to be promoted, there's going to be a ton of glucose in the blood. Insulin, that peptide hormone that we talked about a second ago, is going to help these glucose transporters, or GLUT for short, move this excess glucose into the cytosol of these cells. Now that glucose is inside the cells, we can run it through glycolysis to produce pyruvate. We're also going to produce some ATP and electron carriers as well. Once we have produced these pyruvate, we can either go two different directions. The first one that we're going to start with is whenever we have oxygen present, and we can prioritize the efficiency of ATP rather than the speed. Again, we'll talk about that here in a few minutes. We're going to take this pyruvate and we're going to ship it into the mitochondrial matrix. Just as a reminder, Pyruvate is a 3-carbon molecule. Glucose is a 6-carbon molecule, so we'll be producing 2 pyruvate per glucose. Once this pyruvate is in the mitochondrial matrix, we can send it through the PDC, which is going to form acetyl-CoA. And once we have this acetyl-CoA, we can then start the Krebs cycle. All of this in the aim of producing ATP or electron carriers. So from here, we're going to take all of these electron carriers that we've produced in all of these other processes. We're going to send them through the electron transport chain, where NADH is going to drop off its electrons at complex 1, FADH2 is going to drop off its electrons at complex 2, and then the electrons will be used by the complexes to pump protons out of the mitochondrial matrix, essentially in doing so, creating a really strong proton gradient. There's going to be a lot of these hydrogen protons in the intermembrane space that are going to want to move back into the matrix, but that inner mitochondrial membrane is not letting it. So the ETC is going to be what's creating this gradient of protons, and ATP synthase is going to allow 4 of these protons back into the mitochondrial matrix. This is a favorable movement that ATP synthase will couple to the formation of 1 ATP. This is also called chemiosmotic coupling. We are coupling the favorable movement of an ion, specifically hydrogen protons, down their concentration gradient into the matrix. That's why it's called chemiosmotic. And we are coupling that to the production of ATP. So this is the main way that our body can produce ATP in a very efficient way. It's not super fast, but it's very efficient. We're producing a lot of ATP per glucose. But it does, however, also require oxygen to be present. And there might be some circumstances when our cells do not have enough oxygen. For example, really intense exercise. We might run into a scenario that we don't have enough oxygen in our muscle cells, so they can't really do all of this stuff that's going on in the mitochondrial matrix. So instead of shipping that pyruvate into the matrix, we're going to leave it in the cytosol, where we're going to send it through fermentation. This is what we call anaerobic respiration, meaning it's missing oxygen. So we're going to take this 2-pyruvate, we're going to turn it into lactic acid, and then we can recycle this lactic acid back into glucose by shipping that lactic acid to the liver, which will turn it into glucose via a process called the Cori cycle. So basically, whenever our body decides to go through fermentation and anaerobic respiration, we are sacrificing a whole bunch of ATP that we would be producing in the mitochondria, but this process can occur super quickly, and it doesn't require oxygen to be present. It's way less efficient in that we're only going to be producing 2 ATP per glucose, but it basically just provides us with another way of producing ATP. So we are going to take a little bit of a deeper dive into each of these processes. And we're going to start off with glycolysis. Like I said earlier, we're not going to fully dive into all of this information. This is going to be much more so like a quick introduction to all of these processes. So if you are looking for more, I highly recommend going out and checking out my other videos or resources. So for glycolysis, it's important to note, again, starting off with those glucose transporters, these are going to be those proteins that are going to be shipping glucose into the cells via a process called facilitated diffusion. It's a passive form of diffusion. The first glucose transporter that you need to know a little bit about is GLUT4. This is going to be present on pretty much all of our cells throughout our entire body. So it's always going to be on, and it has a super high affinity for glucose. This is another way of stating that it has a low Km, a low Michaelis constant, if you know anything about enzymatic kinetics. Basically, all of this just means that it is always going to be bringing glucose into the cells so that they can produce ATP. So even if there's not really that much glucose, GLUT4 is still going to work. GLUT4 is activated by insulin, which should make sense. Insulin is released whenever we have a ton of blood glucose. So the presence of insulin is basically just going to help us ship that glucose into the cells using GLUT4. GLUT2, on the other hand, on the MCAT, it only shows up on the liver. And it's only going to be turned on whenever we have high blood glucose concentrations. The reason for this is it's only going to be bringing glucose into the liver with the purposes of storing excess glucose. If we have a ton of glucose in the blood, that means it's in excess. GLUT2 is going to be turned on. It's going to pull glucose into the liver and start storing it as glycogen. So because of that, we say that it has a low affinity for glucose. This is a high Michaelis constant. and it's only going to be operational whenever blood glucose concentrations are high. GLUT2 also serves as a sensor for the body to start producing more insulin. So if GLUT2 is activated, that means there's a whole bunch of glucose that needs to get removed from the blood. There's basically a pathway that GLUT2 being activated is going to cause the body to produce more insulin so that the cells know to take in even more glucose. Again, just emphasizing the fact that insulin's job is just to remove glucose from the blood. That's the only thing insulin really cares about. It sees a whole bunch of blood glucose and it just wants to get it out. This is occurring in the fed state. It's also called the postprandial or absorptive state, meaning that we're mainly going to be doing glycolysis when you've just eaten a meal and you have a ton of glucose that you can produce ATP with. Glycolysis is going to occur in the cytosol of every cell. It's considered an anaerobic process, meaning that it doesn't necessarily require oxygen. It's promoted by insulin, high amounts of blood glucose, and low amounts of energy or ATP, which should make sense. We have a whole bunch of glucose in our blood from our meal, and we want to make ATP with it. It is inhibited by the opposite of insulin, a hormone called glucagon, as well as high energy levels, high ATP levels, and of course, negative feedback loops. Basically just meaning that if we have a lot of the products of these reactions that make up glycolysis, we don't need to keep running the reactions. We have enough, so those products are going to go back and inhibit their own formation. There's 3 enzymes that I recommend knowing. I recommend knowing their reactant and product, as well as the number of carbons of each of those reactants and products. The first enzyme is called glucokinase, which exists in the liver and the pancreas, or hexokinase. This exists in all other cells. Same enzyme, just in different types of cells. The next enzyme, which is the rate-limiting step, the committed step to glycolysis, is PFK1. Basically, once the molecule has been sent through PFK1, that molecule will no longer be diverted to any other pathway, like the production of glycogen or the pentose phosphate pathway or anything like that. Once we send it through PFK1, that molecule is now going to be doing glycolysis. PFK1 also has a way of being directly promoted by insulin because insulin will stimulate PFK2, a very similarly named enzyme, to form a molecule called F26BP, fructose 2,6-bisphosphate, which is then going to turn around and directly stimulate PFK1. So not only is insulin promoting the movement of glucose into the cells, it's also directly stimulating one of our enzymes as well. And the last enzyme that you need to know in the chain is pyruvate kinase. This is a reaction that is going to be occurring twice per glucose because we're going to be taking 2 of the 3-carbon molecule phosphoenolpyruvate and forming pyruvate with that. This is happening twice because we're forming 2 PEP per glucose. So the net products per glucose through glycolysis is 2 net ATP, 2 NADH, and 2 pyruvate. So with this pyruvate, kind of like we talked about earlier, there's kind of a few different things that we can do with it. The first is send it through the PDC, the pyruvate dehydrogenase complex. This occurs in the mitochondrial matrix, except in red blood cells. Red blood cells notably do not have mitochondria, so they are only going to be doing glycolysis and fermentation. And these processes that occur in the mitochondrial matrix, we consider them to be aerobic, meaning that oxygen needs to be present. So we're going to be doing the PDC whenever we both have oxygen present and our conditions prefer the efficiency of ATP production over the speed of ATP production. If we send pyruvate through the PDC, we're going to be able to get a whole bunch of ATP per molecule through aerobic respiration, but that's a little bit slower and it also requires oxygen to be present. If we don't have oxygen or we need ATP super quickly, we're instead going to be doing fermentation and anaerobic respiration. So the basic summary of the reaction of the PDC is we're taking the 3-carbon pyruvate and we're turning it into 2-acetyl-CoA. In doing so, we're going to produce an NADH, one of those electron carriers, as well as a carbon dioxide. I want to take a moment to point out here that every single time we're producing one of these electron carriers, NADH or FADH2, this is a redox reaction. I personally hate general chemistry. That's not really what I'm focusing on for the purposes of metabolism. Obviously for the MCAT, you need to understand redox reactions. But all of these dehydrogenase enzymes are going to be the main ones that are doing all of these redox reactions to produce NADH or FADH2. So we're going to be oxidizing pyruvate, which allows us to reduce NAD into NADH. We're going to see a whole bunch of these dehydrogenases whenever we get to the Krebs cycle. We're also going to do a decarboxylation here, releasing a carbon as carbon dioxide. Just as a reminder, this is going to occur twice per glucose, since one glucose is going to form 2 pyruvate, so you can double all of these products if they're asking about the products per glucose instead of per pyruvate. And the main enzyme that's being used is pyruvate dehydrogenase. Just as a reminder, we must shuttle pyruvate into the mitochondrial matrix. And if we compare the PDC and the rest of aerobic respiration to anaerobic respiration, it's going to be more efficient, meaning that sending it down this pathway is going to produce more ATP per glucose, but it does have more specific conditions that need to be met, and it's going to produce ATP at a slower speed. Conversely, if we don't have oxygen or we need ATP super quickly, we can send pyruvate through fermentation and anaerobic respiration. This is going to occur in the cytosol, meaning that it's an anaerobic process, just the same as glycolysis, thus the name anaerobic respiration, not needing oxygen. And we're going to be doing this whenever we either don't have oxygen or we need ATP super quickly, specifically faster than aerobic respiration can provide. So because the electron transport chain isn't going to be working anymore, those electron carriers, NADH and FADH2, are now useless. We don't have any use for them because their only purpose was to help the electron transport chain pump protons. Now that that's all shut down, we don't need NADH anymore, but we do need NAD+, because NAD+ is a reactant in glycolysis. So basically, fermentation is a way for us to recycle the no longer needed NADH into NAD+, so that glycolysis can continue. Basically, we're going to be taking pyruvate and that no longer needed NADH. We're going to be running a redox reaction that's going to reform NAD+, and it's going to reduce pyruvate into lactic acid. This is the product in mammals. This is one of the main reasons that your muscles tend to hurt quite a bit whenever you're working out. But if you happen to be one of those really cool yeast eukaryotes, instead of lactic acid, you'd be producing ethanol. The enzyme that's going to catalyze this redox reaction is lactate dehydrogenase. Notice it's another dehydrogenase that's at the heart of these redox reactions. And then, of course, just to complete the cycle, to compare it to the PDC and the rest of aerobic respiration, it's going to be less efficient, meaning we're producing less ATP per glucose, but it's producing ATP at a faster speed, meaning more ATP per second, and it can occur in a broader range of conditions. But now, so let's continue through the rest of aerobic respiration. We're going to be shipping this pyruvate into the mitochondria for the PDC and the rest of aerobic respiration. Now that we've formed acetyl-CoA through the PDC, we can then send it through the Krebs cycle. The Krebs cycle is also known as the TCA or the citric acid cycle. All of those mean the exact same thing. This is going to occur in the mitochondrial matrix, which also makes it aerobic. And because this is occurring in the mitochondria, it's not going to happen in red blood cells. Again, red blood cells do not have mitochondria. It's going to be promoted by high amounts of acetyl-CoA, which should make sense. We have a whole bunch of the reactants, so we're going to do this process a lot, as well as low ATP. The goal of this process is to produce electron carriers so that we can make ATP. So if we don't have a lot of ATP, we're going to do this process a lot. It's going to be inhibited by low amounts of acetyl-CoA, as well as high amounts of ATP, basically just the opposite of what it's promoted by, as well, of course, as those negative feedback loops of the intermediates. If we have a lot of fumarate, we don't need to produce anymore, so fumarate is going to inhibit its own production. Negative feedback loops. So as far as what you need to know about the substrates, you need to know their names, you need to know the order of the substrates, as well as the number of carbons that each substrate has. There's a helpful mnemonic that says, "Can I keep selling sardines for money, officer?" which just lists all of these substrates in order, starting off with citrate, going around the circle. As far as the enzymes that you need to know, you'll notice in the figure on the right that there's 2 arrows that do not have an enzyme. You do not need to know the enzyme that's going to put this 2-carbon acetyl-CoA together with the 4-carbon oxaloacetate to get us to that starting molecule citrate. You don't need to know that enzyme. You also don't need to know the enzyme that turns citrate into isocitrate. So the first enzyme that you need to know is going to be isocitrate dehydrogenase, which is going to turn the 6-carbon isocitrate into the 5-carbon alpha-ketoglutarate, a redox reaction and decarboxylation that's going to produce NADH and carbon dioxide as well. Alpha ketoglutarate dehydrogenase is then going to pretty much do the exact same thing, producing the now 4-carbon succinyl CoA, a redox reaction and decarboxylation that is also going to produce NADH and carbon dioxide. Basically the same reaction twice in a row, just with a different substrate. We can then take this succinyl CoA and run it through succinyl CoA synthetase, an enzyme that is going to be removing coenzyme A, and we're going to couple that favorable removal to the production of an ATP. Notice we didn't lose any carbons. In fact, we're not going to lose any carbons for the rest of this cycle. We're basically just chopping off that CoA, which is going to provide the energy that we need to form an ATP. The molecule that we're left with is a 4-carbon molecule called succinate. We're going to run succinate through succinate dehydrogenase. Since it's a dehydrogenase, as you can guess, this is going to be a redox reaction. Specifically, this is going to be forming FADH2. This is the only step in aerobic respiration that produces FADH2. And so because it's a little bit special, succinate dehydrogenase actually is also just going to be directly part of complex 2 in the electron transport chain. Complex 2 is the complex that receives electrons from FADH2. And since this is the only step that is going to produce FADH2, we basically just slap both of those together. The product of succinate dehydrogenase is a molecule called fumarate, still a 4-carbon molecule. We're going to be adding a water molecule to fumarate in order to form malate. The way that I always memorized this was, this is the only step that requires water. Water starts with a W. If you turn a W upside down, you get an M. So the formation of malate, which starts with M, requires of water. That might help you memorize this step. But now that we've produced this molecule malate, we're going to send it through yet another dehydrogenase, where we're just going to oxidize malate into a molecule called oxaloacetate. In doing so, we form the reduced NADH. So remember that this is occurring twice per glucose, because one glucose molecule will produce 2 pyruvate, and those 2 pyruvate will go on to produce 2 acetyl-CoA. So the products of the Krebs cycle per glucose is going to be 6 NADH, 2 FADH2, 4 carbon dioxides, and then 2 ATP. Technically, that step with succinyl CoA synthetase is producing a GTP, but GTP is analogous to ATP for our purposes. So that's the Krebs cycle. Now that we have a whole bunch of these electron carriers, we're actually going to start taking advantage of them. The way that we're going to do that is the electron transport chain. Before we talk about how the electron transport chain works, there's a few important molecules that I'm going to introduce. The first one is going to be ubiquinone, or coenzyme Q. Whenever it's in the oxidized form, we call it ubiquinone. It's going to have a ketone on it. Whenever it's in the reduced form, we're going to be turning that ketone into an alcohol, so we change the name to ubiquinol. Highly recommend understanding the naming convention of the two. Ubiquinone when it's oxidized, ubiquinol when it's reduced. Again, it's also called coenzyme Q, specifically coenzyme Q10. I highly recommend being able to recognize a quinone as well. You don't necessarily need to be able to draw it from scratch, but I'd be able to look at a molecule and know that it contains a quinone, which we can reduce into a quinol. The other molecule that I want to introduce is cytochrome C, or Cyt-C for short. Cytochrome C, the important thing to note is that it contains heme, which heme is essentially just iron with a porphyrin ring around it. I recommend being able to recognize a porphyrin ring, as well as knowing that cytochrome C contains heme, which will have one of these porphyrin rings inside of it. Both of these are going to be electron carriers that are going to help us pass electrons in between the complexes. The electron transport chain is going to occur on the inner mitochondrial membrane. And so, of course, this is still going to be aerobic and not occur in red blood cells. It's going to occur whenever we need ATP and we have oxygen present. And so to actually summarize what's going on here, we're going to start off with complex 1, this complex that's in purple up here. Complex 1 is going to receive electrons from NADH. Now that the complex is reduced, it has a whole bunch of energy that it can use to pump protons out of the mitochondrial matrix. Now that it's pumped its protons, the complex has done its job, but it needs to reset itself so that it can receive more electrons from NADH. So it's going to pass on those electrons to coenzyme Q, reducing ubiquinone into ubiquinol, coenzyme QH2. Complex 2 is going to be similar to complex 1 in that it's receiving electrons from one of those electron carriers we talked about in those earlier metabolic processes. But instead of receiving electrons from NADH, it's going to be receiving electrons from FADH2. The electrons from FADH2 are actually weaker than the electrons from NADH. So complex 2 actually is not going to be able to pump any protons, and it's basically just going to then pass on those electrons to coenzyme Q, forming that reduced ubiquinol. Both complexes 1 and 2 are going to be forming ubiquinol after receiving electrons from NADH2 or FADH2. Just the only difference is that since FADH2 is weaker, complex 2 is not going to be pumping any protons. And again, like we talked about from the Krebs cycle, complex 2 is essentially the exact same thing as succinate dehydrogenase from the Krebs cycle as well. So now that we've formed these reduced ubiquinol molecules, we can take them from complexes 1 and 2, and we can send them on to complex 3. So ubiquinol is going to be reducing complex 3. It's going to give its electrons to complex 3. This is going to allow complex 3 to pump its protons, specifically 4 protons per coenzyme Q. And once it's done pumping its protons, it then needs to reset itself. So it's going to pass the electrons onto cytochrome C. Just want to point out as well, if you haven't noticed, everything on this slide is color coordinated. All of the arrows are color coordinated. All of the molecules and complexes are color coordinated as well. So now that we've given our electrons off to cytochrome C at this point, cytochrome C is going to be passing its electrons onto complex 4, the last complex in the chain. Complex 4 is going to be receiving the electrons from cytochrome C. Whenever complex 4 is reduced, it's only going to be able to pump 2 protons. We're basically sucking the remaining energy that we can from those electrons, and we're only going to be able to pump 2 protons at this point. So now that complex 4 has done its job, it needs to reset itself, and the way that it does that is it's reducing oxygen into water. This is also why we need oxygen for all of these mitochondrial processes. And that's also why oxygen is also known as the final electron acceptor. So just to start tracking the path of the electrons from NADH are first going to go to complex 1, then on to complex 3, and then finally on to complex 4. In the process, those electrons will have directly caused the pumping of 10 protons, like we'll talk about on the next slide, will end up producing 2.5 ATP. So we can produce 2.5 ATP per molecule of NADH. The electrons from FADH2 are going to start off at complex 2 instead, and then get passed on to complex 3 and 4, and in doing so will directly cause the pumping of only 6 protons. This is going to correlate with 1.5 ATP per molecule of FADH2. Additionally, I want to note that since we are pumping protons out of the matrix and into the intermembrane space, the pH of the matrix is going to become more basic since there's less protons in it, and the intermembrane space is going to become more acidic. It's going to have a higher hydrogen proton concentration. To talk about how we can inhibit the electron transport chain, since that's an important topic for the MCAT, we can fully inhibit the electron transport chain. We can do this either by shutting down complexes 3 or 4. So, for example, if we shut down complex 3, we're going to have a buildup of ubiquinol, which is then going to inhibit complexes 1 and 2, fully shutting down the electron transport chain. We can also do the same thing, of course, with complex 4 as well. Either one. So if we don't have oxygen, that's going to shut down complex 4 because it's not going to be able to reset itself. And then we're going to have a buildup of cytochrome C. Complex 3 is going to get stuck in the reduced form, so on and so forth. Basically, if you inhibit complexes 3 or 4 in some way, that is going to fully inhibit the electron transport chain. You can also fully inhibit the electron transport chain by also shutting down both complexes 1 and 2 at the exact same time. Conversely, if we just wanted to partially inhibit the electron transport chain, meaning not necessarily entirely shut it down, but just slow down its production, you can do this by inhibiting either complex 1 or complex 2. For example, if you inhibit complex 2, of course that means that FADH2 is not going to be able to send its electrons onto complex 2. But NADH and complex 1 have nothing to do with complex 2, so they're going to be able to still work fine. So that's why inhibiting either complex 1 or 2 will not fully shut down the electron transport chain. It's just going to slow down how much ATP we can produce. To talk about how we actually produce ATP using this proton gradient, this is where we're going to move on to oxidative phosphorylation, which is going to occur on the inner mitochondrial membrane. It's of course going to be an aerobic process then. And again, this is not going to be occurring in red blood cells because they do not have mitochondria. This is also called chemiosmotic coupling. The reason for that is chemiosmosis is the movement of ions down a concentration gradient. In this example, it's these protons, these hydrogen ions. And the word coupling means that we're just pairing a unfavorable movement or reaction with a favorable one. In this example, we are pairing the unfavorable formation of ATP with the favorable movement of protons back down their concentration gradient. This is going to occur whenever we need ATP and have oxygen present. The enzyme that is going to be doing this process is called ATP synthase. It has two subunits that you just need to know a tiny bit about. It has the F0 subunit, which is going to be a giant transmembrane channel. It's basically just a tube that is going to allow only these protons to enter the tube and go from the intermembrane space where they have a high concentration down their concentration gradient into the matrix. The F1 subunit is going to couple this favorable movement of these protons down this tube to the formation of ATP. It's basically going to rotate as these protons move, and that rotation is going to slap a phosphate onto ADP, forming ATP. And just as a reminder, the formation of one ATP comes from the movement of 4 protons. So just like we did on the last slide, we can talk about how the electrons from NADH, which are going to go from complexes 1 on to 3 and then to 4, pumping 10 protons. Because the electrons from NADH can pump 10 protons, and the production of 1 ATP requires 4 protons, that means on average, each molecule of NADH will be able to produce 2.5 ATP per molecule. Conversely, because the electrons from FADH2 are only going to be pumping 6 protons, that means we're going to be producing 1.5 ATP per FADH2. Because ATP synthase directly relies on the proton gradient that's established by the electron transport chain, we can talk about how inhibiting the electron transport chain can affect ATP synthase. So we just talked about the full inhibition and partial inhibition of the electron transport chain. But if we fully inhibit the electron transport chain, that is going to entirely shut down the pumping of protons, which means that we're going to completely inhibit ATP synthase. ATP synthase no longer has protons that it can work with. But if we only partially shut down the electron transport chain, that's just going to decrease the number of protons that we're pumping. But we're still going to be pumping enough protons that we can still have ATP synthase work. ATP synthase will still be working, just at a slower rate. Full inhibition of the electron transport chain will completely inhibit ATP synthase. Partial inhibit of the ETC will only slow down ATP synthase. We can also inhibit this process of oxidative phosphorylation by a process called uncoupling. This is basically this idea that we can also just entirely remove the proton gradient using some other reaction or something like that. So maybe it's a medication, maybe there's something going on in the cell that raises the pH of the intermembrane space into more basic areas. Basically, anything that removes protons from the intermembrane space is going to stop ATP production, or at least significantly slow it down. That's a process called uncoupling that I will note that we can inhibit ATP synthase without touching the electron transport chain. If you touch the electron transport chain, that's going to inhibit ATP synthase in some way. But this process of uncoupling also demonstrates a way in which we can inhibit ATP synthase. We can inhibit ATP production through oxidative phosphorylation without actually touching the electron transport chain. So that's the process of aerobic respiration start to finish. We're going to talk really quickly about how things change in prokaryotes. The first thing that I want to mention is that there's not really any important changes to any enzymes or intermediates that you need to know. Largely, most of the processes are the exact same, just things are going to slightly be different because they don't have any membranes around their organelles, so they don't have to worry about shipping things into different compartments, like eukaryotes have to. This is going to allow them to produce ATP slightly more efficiently than eukaryotes. They're going to be able to produce a full 32 ATP per glucose, whereas eukaryotes are only going to be able to produce 30 to 32 ATP per glucose. So they're slightly more efficient because of this fact that they don't have membranes that they're having to ship pyruvate or NADH over, just as an example. They also have no mitochondria, so all of their processes are just going to end up occurring in the cytosol, all within the exact same compartment. So the other change that this results in is that since they don't have a mitochondrial membrane to work with, they just move the electron transport chain and ATP synthase to the cellular plasma membrane instead of that mitochondrial membrane. The last thing that we're going to talk about with aerobic respiration is just how much ATP we're producing. Just as a reminder, we're producing 2.5 ATP per NADH and 1.5 ATP per FADH2. So throughout both of these tables, you can see what each process is producing per glucose, as well as how much ATP those molecules correlate with. So for example, in the Krebs cycle, these 6 NADH that we're producing per glucose through the Krebs cycle, if you multiply 6 times 2.5, you're going to get 15. That's how we get that number, that 6 NADH is going to turn into 15 ATP. If we add up all of those ATP that come from the electron carriers or just the processes themselves, that's how we get that number of roughly 32 ATP per glucose. I've also added how much ATP we can produce per acetyl-CoA or pyruvate. That's another thing that the MCAT might ask you to know in those gray boxes on the screen. I'll also add a summary of the reactions going down the middle of the page, just as a reminder of what each process is producing. And then, of course, the final thing that I want to note is what's in an asterisk up here. Asterisk. I can't remember how to pronounce that for some reason right now. That those 2 NADH, because they are made in the cytosol, those NADH have to get shipped into the mitochondrial matrix. There's 2 shuttles that are capable of doing that. And one of those shuttles is going to cause us to lose a little bit of ATP production. It's going to require some energy to move that NADH across that mitochondrial membrane in one of those shuttles. So because of that, that's why we actually see a slight decrease in the ATP yield of eukaryotes. Prokaryotes on the right, however, don't have to worry about this. They don't have NADH shuttles for their mitochondria because they don't have mitochondria, which is why they can get the full 32 ATP per glucose. So that's a relatively short, at least in the speed that I talk about things, that's a relatively short summary of aerobic respiration. Again, if you want a full explanation of any of those processes, you can just check out my other videos. I also have notes for everything on my website. That's where you can also find my other resources, as well as the link to my website for tips, tutoring, yada, 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 so on and so forth.